Biological processes at the molecular level determine our health. It's all about what goes on in our cells. Our uniqueness and our similarities, how we grow, how we become ill, when certain genes are turned on or off, and how we heal and get well, are controlled by a single set of instructions. Instructions that appear in every cell in our bodies. These genetic instructions are the DNA on just 23 chromosomes that we inherit from our two parents that, when combined, result in 23 pairs of chromosomes that constitute our genome. However, genes do change spontaneously, even randomly. Genes change both between generations, that is, from parents to child, and over one's entire lifetime. Against this changing backdrop, our everyday behavior and our environment influence and change our genes. In addition, researchers are constantly exploring ways to manipulate genes to make us a healthier version of who we are. Possibilities exist today that were unknown just a few decades ago. Therefore, we can say that our inherited genes are not our destiny. The fields of biomedical research, applied medicine, and public health, all working to improve our destiny, overlap one another, and we benefit from the interplay. A goal is to better understand all the factors that affect our genes and how genes affect health. We can then apply such knowledge to both prevent genetic defects and, through harm reduction, mitigate or lessen the effects of unhealthy mutations. Applied medicine helps one patient at a time. For public health, the patient is the whole population. A pattern of inherited traits was first observed in 1865 by Austrian monk Gregor Mendel, who conducted experiments in his garden with peas of different colors, textures, shapes, and sizes. By means of tables and mathematics, he explained how traits are inherited. However, it wasn't until 1953, almost a century later, that James Watson and Francis Crick identified DNA as the molecule responsible for inheritance, as well as the development and functioning of all living organisms and most viruses. Using X-rays and quantum mechanics, they discovered DNA to be a double helix or twisted ladder, whose rungs contained the code of life. The double helix model is relatively easy to understand, and in less than a decade it was part of the curriculum in many American high school biology classes. Within this new field of modern molecular genetics, Francis Collins isolated a single gene linked to cystic fibrosis, another to Duchenne's muscular dystrophy, and yet another to Huntington's disease, all in the 1980s. But most diseases are not traceable to a single gene. Multifactorial disorders are caused by multiple genes in combination with lifestyle and environment. This makes it difficult to assess a person's risk for inheriting or passing on a complex condition such as heart disease, diabetes, or obesity. One or both parents can be carriers and pass on a genetic defect, sometimes without knowing it. However, some defects are sex-linked, that is, only the female or the male is a carrier. This is how it works. Remember that females receive one X chromosome from the mother and another X chromosome from the father. This combination XX sex chromosome is what makes a female a female. Males receive one X chromosome from the mother and a Y chromosome from the father. This combination XY sex chromosome is what makes a male a male. In the case of Duchenne's muscular dystrophy, or DMD, the defect is on the female sex chromosome, the X chromosome, so only a female can be a carrier, but almost never inherits the disease. However, if a male offspring receives an X chromosome with the defect from the mother and a Y chromosome from the father, he inherits the disease from the mother. In fact, the male offspring of a DMD carrier has a 50% chance of getting the disorder. DMD is a severe disability that usually results in death at about age 25. It's usually not observable until about age 5, when the child's muscles begin to weaken, beginning with the legs and eventually affecting the heart and neurological system. Such severe genetic disability in one member of a family can and does lead to hardship for the entire family. Some carrier couples knowingly accept uncertainty and fate. Others go to great lengths to avoid the defect. To avoid giving birth to a child with DMD, a couple has several choices. Avoid natural conception and consider adoption. Conceive, and if the fetus is a male, test for the defect, and if positive, terminate the pregnancy. Choose artificial reproductive technology, ART, together with pre-implantation genetic diagnosis, PGD. This involves harvesting eggs and sperm from the parents, artificially inseminating many eggs in the lab, testing the young 8-cell blastocysts for the defect, and transferring only the healthy embryos to the mother's uterus. This method is expensive, is discriminatory in that it is not available to those who can't afford it, does not have a good success rate, and is emotionally stressful to couples, and has moral and religious overtones. Consider this. Can the resources for costly treatments that benefit the few be better used in disease prevention for the many?